As we continue our discussion of genetics, let's talk about how traits can be tracked. It's not always easy to follow human traits because we don't necessarily know the genotype of every individual, but we can figure some of that out by using something called a pedigree. Okay, so when you talk about simple dominant recessive inheritance of alleles, okay, the recessive phenotype is always going to result from the homozygous recessive genotype, but the dominant phenotype can be either homozygous dominant or heterozygous. When you look at traits in nature, the more common traits are not always necessarily specified by dominant alleles. For instance, when we talked about the peas that Mendel was crossing, how many of you have bought peas that were yellow? Not usually are they yellow, they're usually green, even though the green color is recessive. So we're going to look at how we can track that. All right, here are some traits that are found in humans around the world, okay? And the ones on the left are dominant, the ones on the right are recessive. So freckles are dominant, but how many people have freckles? Do more people have freckles than not? No, more people don't have freckles. Same thing with a widow's peak. The widow's peak is when the hairline comes to a point like this. Okay, it can be as severe as this, or it can be a less of a peak, but it comes to a peak rather than being straight across. And again, widow's peak is the dominant characteristic, the dominant trait, but it is less common in the public than straight hairline. And the same thing with earlobes. Free earlobes are dominant, attached earlobes are recessive, and attached earlobes oftentimes are more common than free earlobes. So just because something is dominant doesn't mean that it's necessarily more common in the population. We can look at the inheritance of human traits that follow Mendel's laws by looking at a pedigree. Pedigrees are diagrams that show how a trait is inherited through multiple generations, and it can be used not only for humans but also for dogs and horses and all different kinds of organisms. Okay, it demonstrates the dominant or recessive inheritance, and it can also be used to figure out with the genotypes of different family members. Okay, here's a typical pedigree. This one happens to be for earlobes. Okay, and notice, remember we said that the attached earlobe is recessive and the dominant earlobe is free. Okay, so when we look at the two sisters right here at the bottom of the diagram, okay, this one has attached earlobes, which is the recessive condition, and this one has free earlobes, which is the dominant condition. Okay, they came from two parents who both had free earlobes, but apparently here both parents were heterozygous. And if we look back to the grandparents, the father's parents here were both heterozygous. The mother's grandparents, the father had attached earlobes and the mother had free earlobes but was heterozygous. We know which one is which because of the shape of the structure. The square shapes are males and the round shapes are females. When you're at any condition that you're looking at, whether it is just a trait or perhaps a medical condition, the one that you're looking at, the affected individuals of the traits you're looking at are going to be shaded in. In this case, they're light gray, sometimes they're black, or something darker than the light gray that's shown here. So you can see in the father's family that he has two brothers that had attached earlobes, but a sister that had free earlobes, and we don't know whether she has a homozygous dominant or heterozygous because there are no offspring shown here. Okay, we know that he's heterozygous because he has a daughter that has attached earlobes, and the same thing here for the mother. In humans, there are a lot of inherited disorders that are controlled by single genes, and there are two different kinds of inheritance of those disorders. The recessive inheritance is going to require two recessive alleles to show the disease. We call the heterozygous parents carriers of the disease-causing allele, and the probability of inheritance of that recessive condition increases with mating between close relatives. It's not necessarily common nowadays, but in some cultures and in past times, it might be more common to marry a cousin than it is today. Dominant inheritance of an inherited disorder only needs one dominant allele to show the disease. Some of these are lethal conditions, and oftentimes the lethal conditions are usually eliminated from the population because they don't get passed on to the next generation because the person who inherits that passes away. So here's how this would be inherited, some condition. In this case, we're talking about deafness, okay? So both mom and dad have normal hearing, but they're both heterozygous, okay? So dad's alleles are here. He has either a dominant, his sperm cells have either the dominant D allele or the recessive deafness allele. Same thing for the mother. When we do the cross, what we find is of their children, we have a 1/4 chance of having a homozygous dominant normal. We have 2 out of 4, or 1/2 chance of having normal carrier offspring, and we have 1 in 4 chance of having a deaf child. And so that's how those recessive traits can be passed on. There are several fatal genetic diseases that are present in the population. The most common one in the United States is one called cystic fibrosis. Cystic fibrosis has to do with, is basically a breathing difficulty. It has to do with thick mucus secretions. This is a recessive allele, and it's carried by about 1 in every 31 individuals. There are some other dominant disorders that are present in the population. They include achondroplasia, which is a dwarfism trait, and Huntington's disease, which is a degenerative nervous system disorder. Here's a list of some autosomal disorders that are found in humans. This is in your book in Chapter 9. And so you can see the various things here. We have albinism that we've talked about in class, galactosemia, which is an accumulation of galactose in your tissues, and it leads to mental retardation. Tay-Sachs disease, sickle cell disease, and so forth, and the dominant disorders, achondroplasia and Huntington's disease, and one type, the familial Alzheimer's disease, which is a rare form of Alzheimer's disease. We don't yet know what the incidence is in the population, but it is not generally very common at all. Here is an example. This doctor has achondroplasia, which is a type of dwarfism, and this doctor actually specializes in the treatment of bone disorders in people with achondroplasia. All right, let's go on and talk about there are sometimes that Mendel's laws don't actually seem to apply. There are variations or differences in the type of inheritance, and so we're going to look at some of those variations of Mendel's laws. So sometimes we have something called incomplete dominance that occurs. When Mendel was doing his crosses, he was working with peas, and all of his peas illustrated what we call complete dominance. The characteristics were dominant or recessive, and there was nothing in between. There are, however, some conditions, some genes in which the hybrids in the F1 generation have an appearance or an expression of the trait that falls in between the two phenotypes of the parents. This is called incomplete dominance. Okay, in this case, you have neither allele that is dominant over the other, and both of them are expressed. They both show up. An example is in snapdragons, which is a type of flower. Snapdragons have two main kinds, true breeding kinds, red snapdragons and white snapdragons. And so you would assume, as usual, that the red would be homozygous dominant and the white would be homozygous recessive. When you cross them, what you find is that all of the offspring that are produced from that first cross in the F1 generation are pink. Both the red and the white gets expressed, and so it's kind of like if you take a bucket of red paint and a bucket of white paint and mix them together, you're going to get pink. When these produce gametes, half of the gametes would have the dominant allele and half would have the recessive allele, and you end up with, in the F2 generation, 1/4 of the offspring having red flowers, 1/2 having pink flowers, and 1/4 having white flowers. So this is a very good example of incomplete dominance. This isn't the same thing as the blending hypothesis that people originally thought before Mendel's work, because those original phenotypes actually reappear in the second generation. One example of incomplete dominance in humans is hypercholesterolemia, and that's when you have high levels of cholesterol in your blood. If you're heterozygous for that condition, then you might have high cholesterol, but it's not as high as it would if you're homozygous dominant, recessive rather, so, homozygous dominant. So this is a person who's homozygous for the ability to make LDL receptors. That's the normal condition. So you've got LDL stands for low density lipoproteins, which is what cholesterol falls in. And so there are plenty of receptors for that, and so that keeps the blood cholesterol level low. In the mild disease, you're heterozygous and you have some receptors for cholesterol or for the LDL, but not as many as you do in the normal condition. So you might have moderately high cholesterol. In the severe disease, you don't have any LDL receptors, and so you end up with a very high level of cholesterol in the blood. That's an example of incomplete dominance in humans. There are others, and we'll talk about some of those in class. Another condition that can occur that doesn't really follow Mendel's laws is when you have multiple alleles in the population. Individuals only carry 2 different alleles for a particular gene, but oftentimes there are more than 2 in the wider population. And a really good example of this is human blood types. The ABO blood group has 3 different alleles for the single gene. Each person can only have 2 of those alleles, and as a result, there are 4 possible human blood groups, the A, B, AB, and O, and they result from different combinations of these alleles. The A and B alleles are both expressed in someone who is heterozygous, and this is called codominance. They both are dominant and they both show up. Okay, in codominance, neither allele is dominant over the other. Both alleles are expressed and they're observed as distinct phenotypes in that heterozygous individual. And AB blood type is an example of that. Let's see how it works. If you have blood type A, okay, there are 2 possible genotypes that can produce blood type A. The convention, or the common use of the symbols for this is using the capital I as the dominant allele, and this with the superscript of the A or B type, okay, and then the lowercase I is the recessive allele, okay. So if you have type A, what that means is that you've got the A type carbohydrate on your red blood cells. If you have type B, you have the B type, and you can see in the picture here, the way it's illustrated, there are different carbohydrates. If you have AB, then you have both types of carbohydrates on your blood cells, and if you have type O, you have neither one. Now, what effect does that have on your blood? Well, it only affects if you mix it with another kind of blood, okay? And so because you have antibodies in your blood for the opposite of the blood type that you have, and antibodies will cause clumping of the blood cells, which can cause blood clots and other kinds of serious conditions if the bloods get mixed within the body. So if you have type A blood, you have anti-B antibodies, and if you mix the type A blood with the different blood groups here, you'll see if I mix A with O, it doesn't cause clotting because O doesn't have any of those carbohydrates on its surface. Type A, no problem, because the anti-B antibodies don't work against the A antigen. But if you mix it with type B or type AB, you do get clumping or clotting, agglutination of the blood cells. Same thing happens with B blood. B blood has anti-A antibodies, which will work against type A and type AB because both of those have the A antigen. Type AB blood doesn't have any antibodies against either one of those because it has both antigens present on its blood cells, and so therefore type AB blood can receive blood from any donor. And then type O blood has both antibodies because it doesn't have either one of the antigens, and therefore the only type of blood that a person with type O blood can have is type O blood. And we'll talk more about how this works in class, in reality. There's also a condition called polygenic inheritance. This is when you have a single phenotype that results from adding together 2 or more different genes. Human skin color is an example of polygenic inheritance. So is eye color. And we'll talk about a couple of other things as well in class. Okay, so here we have skin color. Skin color has 3 pairs of alleles, 3 genes that code for the production of melanin, which is the pigment that colors our skin. This is different from the very, very light, which is homozygous recessive for all 3 genes, is not the same thing as albino because they do produce a melanin, they just don't produce as much melanin, whereas someone who's albino doesn't produce any melanin at all. So we have the very light with the very dark, okay? And so this would be dominant for each of those 3 genes. When they're crossed together, the offspring that are heterozygous for both, for all 3 genes, would have an intermediate color of skin in between. And so here's what you see when you cross them all together. You have a total of 64 possible combinations, okay? And so you end up with 64 total crosses here. This is a tri-hybrid cross, and so you'll end up with, in your population, 1/64 with very, very light, 1/64 with very, very dark, and all the different colors in between, which is why we see so much variation in skin color among humans. We can graph it like this and show what the fraction of the population is, and you'll see that it forms this bell-shaped curve, which is called a normal distribution, and that's commonly seen in traits that are coded by multiple genes. There are also characters that can be affected by the environment. Skin color is one of those. Again, okay, skin color, as you know, is affected by exposure to sunlight, and there are also some other things like susceptibility to diseases like cancer. There are differences between identical twins and things like that. The genetic influences are inherited, the environmental influences are not. So here we see several friends with different colors of their skin depending on their exposure to sunlight as well as their genetic inheritance. And here we have 2 identical twins that, as you can see, are very identical in terms of their facial appearance, but their skin color is very different just because of their exposure to sunlight.